Hello, everyone. My name is Ming-Li Wang. I'm from Hawaii Agriculture Research Center. I would like to update our progress on coffee breeding for coffee leaf rust resistance. Before I get into it, I'd like to give a shout out to Hawaii Department of Ag. The reason is coffee leaf rust has been around the world for more than 100 years. And between the last outbreak in Jamaica in the 80s, it was 35 years before it actually came to Hawaii. So whatever HDOA Plant Quarantine Division is doing, it's doing a good job. It works. And the Hawaii and the coffee leaf rust race that came to Hawaii has been determined to be race 24. And so far, we only know we have one race. However, in the world, there are more than 50 different coffee leaf rust races. And on the plant side, there are at least 11 or more resistant genes involved. So the interaction between the fungus, the pathogen, and the host plant, coffee plant, are very complicated. And it involved multiple genes. And this cultivar may be resistant to our race of CLR, like 24 now, but it may or may not be resistant to any other CLR race that could come to Hawaii in the future. And to manage CLR, we need to integrate a whole series of approach in order to be able to manage it. That includes surveillance, sanitation, pruning, plant nutrition, fungicide application, and the use of resistant cultivars. And in order to obtain CLR resistant cultivars, we can import that the resistant variety that already been created somewhere else in the world, or we can breed locally adapted cultivar with CLR resistance in order to maintain the quality. Imports is very important. However, it has the issue of long quarantine time and intellectual property right issue. And it may or may not be adapted to Hawaii's growing environment. And it may also change the quality of our coffee. So it's still important to have our own breeding program. So at HARC, the breeding program was initiated by Dr. Chifumi Nagai in 1997 with the help of Dr. Bob Osgood. The goals at that time is to develop our own cultivars with unique beverage quality in anticipation of a future CLR infestation. At the peak, HARC's germplasm collection has more than 700 genotypes of coffee plants and more than about 3,000 trees. And currently, we have 465 genotypes and more than 1,800 trees still in the field. And we have, in 2019, we have started a transfer of these germplasm collection to Tracy Matsumoto at PBARC. Currently, we have 2 breeding programs at HARC. First, the larger project was funded by USDA, and this is so basically we want to develop a breeding pipeline to screen for CLR resistant cultivars and maintain unique quality. I will be responsible to create potential CLR resistant coffee seedling, both continue from Chifumi's breeding program and also bringing new source of resistance. Dr. Lisa Keith at PBAR will be responsible to screen those coffee plants for CLR resistance, and genotyping, the DNA typing, will be responsible by Dr. Dapen Zhang, also ARS in Maryland, Beltsville. Dr. Qingyi Yu at PBAR will develop molecular marker for CLR resistance. And Tracy will be responsible for phenotyping, including cupping quality. And Andrea from UH will be distribute any information that's useful that coming out of this project. A second and smaller internal HARC project is to try to combine nematode resistance with CLR resistance. And I'll talk about a little bit more later. I'm going to talk about the CLR resistant effort first. Among HARC's germplasm collection, we have 3 different sources of CLR resistance. Both Cartimoa and Obata, the resistance came from Timor hybrid, but they came from 2 different selection of the Timor hybrid, A32-1 versus /2. And we have a third source of CLR resistance, which contain the SH3 gene from Liberica coffee. However, we discovered this BA10 later, and so we haven't started using this genotype yet. I'm going to focus my talk on Cartimoa and Obata. For Cartimoa crosses, Dr. Chifumi Nagai already made crosses with from these 2 different selection of Cartimoa, 5175, 8667 are both Cartimoa. She made crosses between these Cartimoa with locally elite selected genotype, Hawaiian commercial variety. In 1999, she already made the original cross. And she also made 1 field-grown population that advanced 1 more generation between a Cartimoa and a Mocha type. So this is the genetic resources we have when we start the project. The next few slides I have shown in last year's update, so I'm going to go through it fairly quickly. This is a list of all the plants all the crosses Chifumi made in 1999, and I separate into 5 different groups: Mocha group, other, red Cattleya typica, and yellow. And here, this column shows the female parent versus the male parents, and MA represent Maui, meaning this genotype was originally identified on Maui. OA for Oahu, so it was found on Oahu. KA is Kauai, KO is Kona. So she went around the island, found the elite trees, and bring the cell-pollinated seeds or cuttings back to HARC's Kunia station and made crosses. And from the F1 plants, we identify several individual of F1 plants from each cross, and then cell-pollinated and created F2 population. And we have more than 3,000 F2 individual from these 15 crosses. And the top one is the population I mentioned earlier that we already had grown them in the field. They were originally for genetic mapping. But we're using, since that one is more advanced, we're using that population to test if our strategy will work. So this is the genotyping, DNA typing result. And green represent the DNA marker from Cartimoa, represent DNA marker from Mocha parent. And indeed, every individual has some marker from each parent at a different ratio. And also, Dr. Lisa Keys did the initial CLR bioassay on this population. And in this chart, the higher the bar means the more CLR reproduction, the more spore produced. And we did find some individual that never produced spore. So those are the ones we were particularly interested. We actually identified 11 F2 individual, and this is the leaf of the individual, what it looked like. Get this out of the way. Anyway, some of the individuals look like the Mocha parent, some look like the Katimora parent. Same thing is, we can observe the same situation with the cherry. So Mocha cherry is shorter, this dimension, versus normal cherry. This is a Katimora parent. I put a red Cattleya grown from the same field just for comparison. And some other cherry look like the Mocha parent, and some look more like the Katimora parent. This is as expected. And last year we reported we were able to collect sufficient cherry from 25-19 for cupping. And this is the cupping results. However, these were grown in Cunha, and it seems like everything that we collected, even the Katimora and the individual, they kind of normalize the cupping score. So we don't think this Cunha is a good location for field trial. So we try to do the similar thing with Obata. So in 2022, we made several crosses between the same Hawaiian elite parent with Obata. So these are the ones, again, other type, or in this case, they are actually in the similar to Mocha. Red Cattleya, Typica, Yellow Cattleya. We also including the some Ethiopian wild, semi-wild accessions, because these were nematode resistant. So this is the result of a nematode pod test. So the Ethiopian wild semi-wild accession the nematode cannot multiply, reproduce in the soil that's planted with the Ethiopian semi-wild genotype compared to the susceptible Kona typica and Yellow Cattleya control. And we also got some of the Ethiopian semi-wild accession cup tested, and they are kind of okay, at least similar to SO28. So that was old story. I already talked about that last year. So this year we're focusing our effort on CLR bioassay. And Lisa had always just mentioned, in one sentence, we tested with bioassay, and the result is this plant is resistant or not. However, I didn't realize there's so much effort involved in this one sentence. We sent two people, Sayaka and Nathan Fumiya, to PBAR to bring the technique back to HARC so we can increase the throughput and to screen the plants faster. So this is one of our results. This particular one, KO34, it's a typica. You can see the yellow lesion and the necrosis occur. And in a close-up look, you can see the yellow, or orange actually, orange spore produced, versus Katimora has no, in this case, it didn't even have much lesion. Maybe here's the lesion, but there's definitely no spore. So this is one of the F2 individual, number 1733, and it looks similar to the typica. It also produced spores. So this is a susceptible individual. We throw that plant away. And there are some plants look like this one, has some lesion, but never produce spore. When they have lesion, we call them tolerant. When they don't even have lesion, we call them resistant. So since we spend most of our time on CLR bioassay, I have to tell you how difficult this whole process is. There are two major rate limiting factors for this assay. Why does it take so long? First, it's availability of spores. I believe Blaine in Lisa's lab had to drive from Hilo to Kona to collect spore. For us, at least, we have a whole population of wild coffee tree in our Maunawili station that's heavily infested. However, if the weather is not right, it's too dry, there's no infection, not much infection, then there's nothing to collect. And if the weather, if there's a big rain that wash all the spore out, no, we still have no spore to collect. So basically, Sayaka folks has to hike through these wild woods, no trail. She actually got lost once, and then had to, they hike through it with a machete and collect leaves, bend down these long skinny plants, collect leaves that are infected. This table of leaves probably will give us enough spores to inoculate 50 plants. And from these leaves, we use a little razor blade and scrape the spore into the little tube, and we add water and inoculate the leaf disc, as I showed you earlier. So it's difficult to find enough spores sometimes. So when we can find a lot of spore, we try to do a lot of inoculation. The second rate limiting factor, we need to have space to incubate these leaf discs after inoculated for 4 to 6 weeks under the condition that's optimal for the fungus, not for the plant. As you can see here, we cover the light fixture to reduce the amount of light, because strong light will kill the fungus spore, prevent the infection from occurring. We also instead of trying to do it in Petri dish, now we are trying to do it in 96 well plate to save space. And we need to incubate for a long time, 4 to 6 weeks. Okay, so far, we have identified 39 resistant plants. That means no lesion, no spore. And 308 tolerant plants. That means some yellow lesion, but no spore. And 414 susceptible plants. That means they produce spore. These plants are discarded immediately. And we still have almost 800 plants in the pipeline. This number changes every week, by the way. So we still have a ways to go, but at least I can see an end coming. This is what the plants look like. They've been in these little pots too long. They really should be in the ground or at least in the bigger pot by now, but I don't have space for them. And once they are if I transfer them into the bigger part, it will be harder to ship, because the field trial will be done in 4 different farms on 4 different islands. Greenwell Farm in Kona, Maui Grown on Maui, and Oahu is Doe Foods or Wailua Estate Coffee and Chocolate, and in Kauai Coffee. So once they're in the ground, we'll take the phenotypic data, and that will include all the agronomic performances, and especially, most importantly, cupping quality. But before we get to cupping quality, we will do metabolomic analysis to try to predict, because there are already predictor of chemicals identified that can reduce the number that we actually have to cup. If we're able to select a good performance and good quality CLR resistant plant, because they're in the F2 generation, if you take seeds from those plants and plant it out, the DNA will be segregating, so they will not perform exactly the same as the mother plant. They will need to be vegetative propagated. We do have the technology and experience on coffee micropropagation through somatic embryogenesis. Right now, we have Obata in the pipeline only. These are the people involved in the research. As you can see, there's a lot of people spend a lot of time on the CLR bioassay and the field maintenance and the greenhouse maintenance. And on the right side is my collaborators. Thank you very much.